Yay! Do you know what topic 1.6 is? It's our last topic in topic 1. Woohoo! Cells. We'll never talk about cells again. Just kidding, that was a lie. All right, cell division. I am hopeful that you have some memory of mitosis, but that's okay if not. You should have a handout in front of you that I have given you, and hopefully I remember, so that you are taking more with the diagrams and looking at. We're going to look at how, why cells divide. And then 1.6 part 2 is going to be looking like all the things that go wrong during cell division. Woohoo, not right. I'm not going to talk about things that go right. I'm talking about things that go wrong. Okay, topic 1.6, cell division. Here goes. Okay, here's what we're going to be looking at. Like I said, in part 1, we're going to be looking at mitosis and the cell cycle and all the parts of interphase, protophase, metaphase, telophase, anaphase. It's mitosis. We're going to talk about cytokinesis, when it makes new cells. We're going to talk about how cells regulate themselves, why they die, and again, when things go wrong. Also, microscopes. Yay, some things you've already looked at. Okay, let's start with the cell cycle. The cell cycle, where will my head cause the least amount of? Oh, there we go. Okay, cell cycle is looking at a cycle, something that starts and ends back where it began. But looking at, like, here's the cycle it goes through from I am a cell, and now I'm going to divide and become 2, right? It's going to go in this order. You need to know the order, and then you need to know the things that happen in each and what needs to happen in order for those things to happen, okay? So IMPMAT, IMPAT, interphase, metaphase. Interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Those are going to be our order. We'll talk about some ways to remember it, but interphase takes up 3/4 of the cell cycle, right? So you can see this little green area. Here's everything that's in interphase. It's basically getting the cell ready to divide. You should know that the cell won't divide if it doesn't have enough resources. So if it's not getting enough food, right? If it's not getting enough oxygen, it's not getting what it needs, it doesn't have space, it's not going to divide. But basically it's like, hey, we're moving, we're becoming 2 different cells. We're going to spend 3/4 of our time packing and getting everything ready. And then that last part's when we actually move, right? If you've ever moved, this is pretty much how it happens, right? Lots of preparation, little time moving. Okay, so within interphase, which is our first phase here, getting ready, you've got G1, S, and G2. G stands for growth, right? So I get growth, replicating, and then more growth, right? So G1, what happens is I've got growth and metabolism, right? So I'm looking at the cells starting to get bigger, the metabolism, which is what's those chemical reactions are starting to change, and it's getting things ready for the S phase where DNA is going to replicate. Okay, so now I've got 2 sets of DNA, yay! And then I'm going to keep preparing, right, and proofreading, making sure all the DNA I made is correct, because if there's anything that goes wrong here, it's not going to divide. Like if it goes back and it's like, you didn't copy that right, the cell's not going to divide. So these are kind of checkpoints too, we'll talk about. And that all prepares us for M phase, which is the mitosis phase, right? So I've got interphase getting ready, and then I've got the mitosis phase, what we'll talk about in the next one. Okay, cytokinesis is where it actually splits in 2. Cytoplasm, right? Kinesis moving apart. All right, interphase and M phase. So here is more information on interphase, right? So my G1, S, and then G2. So you can remember that the doctor, all these mnemonic devices, right? So my mnemonic device is doctor. The DNA replicates, right? So I get that during X phase. I also get all the organelles that duplicate. So if you were to outline what happens in interphase, doctor, DNA replication, my organelles, I just said this all, but so you can like kind of mnemonic device next to it. The cell's going to increase. They need to get bigger because all these things are being packed up. The proteins and the DNA. So DNA is copied. I'm going to double all my proteins that I need to take with me. I need all the sufficient nutrition to make sure that I can. And then respiration, which is ATP stocking, are needed for division process. So I'm going to make sure that I have all the energy that I need. So I'm getting everything ready. DNA, organelles, cell growth, transcription, obtain nutrients, and then respiration. Okay, so I'm getting ready. All right. The S phase, here we go, of interphase, where my DNA, there we go. Here we go. Yeah. Is where my DNA is replicating, right? So a little bit more on that. What happens to my DNA? Because DNA is coiled so tightly around itself. Oh gosh, what were we talking about? Were we looking at this in class? Right, where it's all tiled and it's in on itself because it needs to be super compact, right? It's spun into 2 strands going that. It's loosely packed, right, with chromatin in the nucleus. So it's kind of like all over the place, right? So like this is how DNA is before it's moved, but usually like it's kind of like this. So what happens, it's kind of like loosey-goosey all around before it divides. And then mitosis, because I need to divide, right, to copy and divide, it tightens into chromosomes. Like it becomes these very visible, very tightly packed chromosomes. It super coils, right? So super coils around the chromatin, so it gets all tightly packed. Chromosomes, as you know, are particular to certain living things. So I get, humans have pairs of 23 pairs of chromosomes. So 46 is our chromosomal number. So the number of chromosomes that occur, like that is created during this phase, is specific to all different organisms. Okay. What happens during that time is my single chromatid, right, which is half of a chromosome, is going to partner up and put down my DNA here. With a sister chromatid, right? So I've got one little thing and now I've got 2. Yay, post doubled, right, during S phase. Okay. Now I'm prepared. I've got all my DNA, I've got everything ready, and now I'm going into mitosis, right? That's that last phase. So mitosis is the prophase, metaphase, anaphase, and telophase. These are the different phases that you'll need to know. So go ahead and sketch them into the boxes, right? My pro meaning 1, pro means 1, right? So I've got them all. They're all sistered up, they're paired up, they're condensed. You can see my nuclear envelope right here. It starts to disappear because the nucleus is going to go away, that membrane. And then in metaphase, they all meet. They met in the middle, right? So they all meet in the middle here. And then in anaphase, right, they start to come apart, my sister chromatid. So I've got double here. They start to come apart. They are pulled right back to where they're going to be their new cell. And then in telophase, I get what happens is I start to see that membrane start to form again, and I'm just about to go into 2. Okay, so telophase, we kind of look like, well, you guys don't even know what an old telephone looks like, but it kind of looks like that, right? So telophase. So the division of the cell occurs concurrently with telophase, right? Telophase and then cytokinesis. Okay, now a little bit more. So adding to those, so that was the general. Make sure for prophase that you have these ones down, right? So that I just said that nuclear membrane breaks down. My pairs kind of start moving to the poles, right? I get to see, this is where I actually get to start seeing it under a microscope because it's all super coiled. Metaphase, the spindle fibers connect and they bring them to the center. They met in the middle, right? So the microtubule spindle fibers, those are the ones that are connected to the centromere, which is the middle. It's kind of like attaches there and it's going to start pulling them this way, and then the opposite for the next one. So the spindle fibers contract, causing it to align in the center. Okay. Anaphase, then I get the spindle fibers contract to cause them to break apart, right? It's kind of like Spider-Man, no? Okay. And they move them to the opposite poles, right? So I'm getting those fibers, they disconnect centromere, chromosomes start to decondense, so it goes back to the singular. I get new nuclear membranes forming, and then 2 identical cells, right? 2 identical to the mother. Okay. Here's cytokinesis. It looks a little bit different from animals and plant cells, so make sure you write that down, right? So we've got cytoplasmic division here. Cell splits into 2. So the microtubule filaments, which form the ring right here, right? It kind of contracts together and creates the 2. Versus in plant cells, the vesicles form at the center and they fuse here, right? So it's almost like this is almost like pinching something into 2. This is almost like vesicles, they form at the center to create an end plate and then they separate. So they almost like build something new and then separate, right? So this cell separation is called centripetal, right? So centripetal, and then this is called centrifugal, right? So centripetal, centrifugal. Make sure you know those 2 things and those are written down. Okay. All right. So here's a summary of mitosis. You can see it happening here, right? So just watching I'm going to get rid of the sound though. Here you go. So as it moves, you can see the development starting to get bigger, right? Woohoo! You can see my prophase. They are visible. Attach my spindle fibers, met in the middle and metaphase, pull sister chromosomes apart, chromatids apart, get anaphase, and get new nucleus, and then I get it split into two, cytokinesis. Okay. Now, meiotic division happens in my somatic cells, my body cells, right? The only place it doesn't happen is in your sex cells. That's meiosis. So mitosis occurs in somatic body cells. It involves just a single division, right? It gets more complicated as you move on. Two identical daughter cells to the mother cell that are genetic clones, so creating an exact copy of what happened before. All right. Now, that's going to lead us into, well, let me show the microscope real quick and then I'll do part 2 later. So identifying these under a microscope. We actually looked at the onion root tip before, but we'll look at it again now that you know about these different phases. You will have to be able to identify these on a test and in a microscope, right? So interphase, right? It's just getting bigger and starting to see the nucleus condense. Prophase, starting to become visible. Metaphase, I see it in the middle of the cell. Anaphase, I start to see them pulled apart. And then telophase is when I see the two different cells, right, with two different nuclei, right? This is bigger than this one and this one. This is more circular, and this one is, I can see them moving apart, okay? So pretty cool. You can see those underneath the microscope, and we will practice seeing those in class, okay? Very last thing, remember that we talked about in the very beginning that interphase takes up like the majority of the cell cycle, but this is called the Mitotic Index. So you can actually calculate from the total number of cells in the cells in mitosis, like what the mitotic index is for that part of the plant, the animal, whatever it is. Super simple calculation. I look, oh, look, I've got 20 cells in mitosis. There's 75 cells. So 0.2, 26% of my cells are going through mitosis right now, right? Which should be good, right? 3/4. So you can select the picture to view the cells with visible and mitosis versus those without, right? So you can actually count the number in anaphase, telophase, mitosis, and calculate the mitotic index. Something else to look forward to practicing in class, okay? All right. What we're going to talk about in lecture 2, we'll keep that for lecture 2 and show, all right, is why we do mitosis and things that can possibly go wrong, all right? More mnemonic devices. Look forward to that. I'll see you later.